Now, what I wanted to show was this particular slide before we go any further, and this comes back to one of the methodologies of GWAS. So if you're thinking about studying for the exam, and as I said, the exam questions will combine methodology with application. The methodology, for example, for the GWAS, what we need to understand is, of course, that we're working on whole genome studies. We are fragmenting the genome and we're hybridizing that fragmented genome to what is called a bead chip, where it can be genotyped. On the bead chip, of course, is short sequences of DNA that correspond to specific SNPs across our genome. And these SNPs are what are called tagging SNPs. And tagging SNPs define haplotypes. And haplotypes are regions of our genome which are defined by common points of meiotic recombination. So all the SNPs within a haplotype are said to be in linkage disequilibrium. Now what we see here in this complicated looking diagram is actually a very simple pictorial way of representing linkage disequilibrium and haplotype blocks. So if we look across the top here, in this region here, in red are shown haplotype tagging SNPs, and in black are shown all the other SNPs across that locus. Here we have a map of linkage disequilibrium, going from no linkage disequilibrium to very, very high linkage disequilibrium. As I said, SNPs that are within the same haplotype will exhibit very high levels of linkage disequilibrium because they tend to be associated together in a population, and hence they are in linkage disequilibrium. What we do is we simply read off, for example, from this first SNP, we read here and here, if these two SNPs occur, or the alleles of these SNPs occur together in a population, then they are coded blue for the high linkage disequilibrium. And for example, you can see here that we have a very, very strong block of linkage disequilibrium. And collectively, this triangle in red makes up one haplotype block, and this other triangle here makes up a second haplotype block. So this is a way of visualizing haplotype blocks in the human genome based on color coding the strength of linkage disequilibrium. So coming back to the methodology of GWAS, what we need to understand is that we can only do these GWAS because we look at a fraction of the 4 million SNPs or so that are across each genome, and we use the several 100,000 tagging SNPs, which collectively identify all haplotypes across the genome, and that these tagging SNPs will give us associations based on loci that occur within these haplotype blocks. Of course, the problem with using these tagging SNPs is that they may or may not be the true causative SNP. In other words, the tagging SNP may cause an amino acid change, or it may not. And in most cases, it actually has no effect. And that's when we need to go searching all of these other SNPs that are in linkage disequilibrium with the tagging SNPs and search for the true causative SNP. So this is where we come back to this concept of what's called direct and indirect associations that we've talked about earlier in the lectures. And so what this comes from a study where, if I just go back a couple of slides, sorry, that one's the one I want. What they've done in this study is that they did an initial GWAS using tagging SNPs. They identified haplotypes. They were unable to identify the causative SNP from those tagging SNPs. So they did a second association study. And what they did is that they designed a chip that contained all of the SNPs around particular tagging SNPs. In other words, they really looked, if we go back, they started genotyping each and every SNP through a haplotype block because they wanted to actually map where the true association was. And so that's what they've done in this particular study here. That really zeroes into a particular region in a haplotype, and it provides the geneticist with a much better idea of where the true functional SNP is. And of course, it's then up to the biologists to do the actual functional test and see whether we have an EQTL and that resides in an enhancer, for example. This is just a picture of type 2 diabetes, a very complex looking table, but the information in it is actually very simple, in that what we see is that all of these SNPs have very low odds ratios, going from about 1.09, 1.08, 1.23. So collectively, tiny risks are singly tiny risks associated with each of these alleles. You can see the risk allele frequency is anywhere between 0.2 up to 0.7. We even have 0.9 in a population. So these can be incredibly common in a population as well. And most of these SNPs actually map to enhancers, and in the case of type 2 diabetes, they map to enhancers that control the expression of genes in pancreatic islet cells. So the GWAS of type 2 diabetes have made perfect biological sense, in that what they're showing is that the structure of these common genetic diseases is many SNPs, each of which has very, very low penetrance or very low odds ratios, and that tend, in most cases, to map to enhancers that control the expression of genes. And in type 2 diabetes, these genes are typically involved in the processing of insulin. So the GWAS of type 2 diabetes have actually been very valuable in giving us an insight into the molecular and genetic basis of this particular disease. Of course, as far as its application to the clinic goes and genetic tests and so on, it's simply not possible given the huge number of these variants and their very low effect on phenotype, as well as the very strong influence of the environment. But what it does do is it gives us information on pathways which could in the future be targets for therapy or interventions into this particular disease. So that's really where we leave, I think, type 2 and type 1 diabetes. But just to summarize, that if you're thinking about comparing and contrasting, what we're seeing is that type 2 diabetes, it's all about multiple alleles with low odds ratios. They map to enhancers, which are typically in the islets of the pancreatic cells. They bind transcription factors, an example of which is FOXA2, and this is a transcription factor which is particularly important in the regulation of islet cell function. And what it's showing is that type 2 diabetes, the genetic risk is affecting insulin production and processing, body mass index, and the beta cell number and formation. If we then look at a comparison with type 1 diabetes, however, we're seeing that that is very much this autoimmune disease with very strong association, very high risk factor with particular MHC genotypes. And that is in interaction with SNPs in genes that regulate T cell activation, such as PTP-N22, and that there is clinical interaction between these SNPs and the MHC, and that was shown in a number of studies already. So two different illnesses, both of which center on the production of insulin, but genetically very different, and in their basis very different. Type 2 diabetes, a metabolic disease; type 1 diabetes, a immune-mediated disease.